So this next section on genetics is going to talk about genetic inheritance beyond what Mendel did. It extends on what he did, but he didn't study these kind of things in his pea plant experiments. So let's just review what the principle of segregation and the principle of independent assortment means. So the principle of segregation is if you have any genotype, let's say that's your genotype, in your sex cells, when meiosis occurs, those alleles will be segregated or separated, one per each gamete that you make. So the principle of segregation, meiosis separates those alleles, one per gamete. 50% of your gametes will get big A, and 50% of them will get little a. And that's why we set, when we do a Punnett square, that's why we don't put A like that. We separate it. Okay? Now, the principle of independent assortment simply states that if you had A, B, talking about two genes, these two genes will segregate in meiosis separately from each other. The way one separates isn't controlled by the way the other one separates. So you would basically, if you were prepare this for a Punnett square cross, you'd have 4 different ways the gametes would have the A and B gene. And we do that by the FOIL method first. AB, outer, A little b, inner, little a big B, and last, little a little b. So the A and B segregate, they separate individually, again by meiosis, and again to the sex cells. So this is not diploid. This is 2 different genes getting mixed up like shuffling a deck. And that's because the chromosomes, when they pair up at meiosis metaphase 1, let's say pink is mom and black is dad's chromosomes that you have in your sex cell. In your normal cells. So that's a germ cell. So let's say that's you. And I'm just going to make 3 different genes. So let's say mom's is also the X gene. And then mom has a recessive B gene. And mom has recessive little C gene. So the way these guys line up at the metaphase plate at metaphase 1 is totally random. All the X's and B's don't line up on one side. They're mixed up. They're independently assorted. The chromosomes are, therefore the genes are. Now we're going to have non-Mendelian cases where the alleles are not completely dominant. They're incompletely dominant. We'll talk about that coming up. A gene could have more than 2 alleles, like blood types. We'll talk about that. A gene can produce multiple phenotypes. So you can have, instead of just 2 phenotypes, you could have 3 different phenotypes, or a blend of all kinds of in-between phenotypes. So we'll talk about that. Okay, in incomplete dominance, the heterozygous genotype will give you a phenotype that's a blending of the 2 other phenotypes. So let's say the genotype for red roses is big R, big R. The genotype for white roses is little r, little r. Okay, and then the heterozygous genotype is big R, little r. But this isn't, if this is incomplete dominance, then there'll be a blending of the 2 traits in the heterozygous. Now, they're saying this is not a blending. Well, they mean like paint. If you mix white and red paint, you'll never get, it'll make pink, and you can't separate later the white and red from it. Whereas if you had 2 pink roses crossing, you will get some red and some white offspring. Let's just do one. What if you had 2 pinks crossing? Okay, you get a red rose there, child. You would get a pink one there. You would get a pink one there, and you would get a white one there. So that's why they say the genes aren't blending together. The proteins that you're forming from the gene, the pigment, red and white pigments, are blending, not the genes. Okay, so what would be the phenotypic ratio if you cross 2 pink roses? It would be 25. Let's do percentages. 25% red, 50% pink, and 25% white. Okay, this is not complete dominance. This is incomplete dominance. Or you could say 1 red to 2 pink to 1 white. Or you could say 1/4 would be red, 1/2 would be pink, 1/4 of the offspring would be white. Okay, what I showed you on the previous page was the F1 generation. So the parental generation is what they're showing here. That would be a red one is crossed with a white one, incomplete dominance. So if you did that cross, you can see that 100% so the F1 generation would be 100% pink flowers. And then if you cross them, if you cross, and here's the cross for doing that. You can see it's really simple. Now if you cross those, you would get what we just did on the previous page, and that'd be your F2 generation. What do we say? 25% red, 50% pink, and 25% white. Okay, codominance is like incomplete dominance. And blood types are an example. Okay, we got 3 blood types. We got type A, type B, type AB, and type O. And it has to do, your blood type has to do with the marker proteins on your red blood cell. Okay, the proteins on your red blood cells. Okay, so if you're type A, you only have A proteins only. So we say the A allele forms the A proteins on the red blood cells of type A blood. Okay, so if you have type B blood, you only have B proteins. So you have the allele for B proteins on the red blood cell, because the allele B codes for the B proteins on the surface of the red blood cell. If you have type AB, you form both A and B proteins on your red blood cell because they're equally dominant. They're co-dominant. It's not. And then if you have type O gene, you will produce, if you have type O blood, I should say, you will produce no surface protein, blood type proteins at all on your red blood cells. So let's make a little chart here. Blood type, phenotype, and then genotype. So if you're blood type A, you have A proteins only. On the RBC. Your genotype, however, could be, okay, you're diploid, okay? This is not sex cell. Your genotype would be either having 2 A alleles, or you could have AO, because O doesn't form any red blood cell protein at all. Okay, if you're type B blood, you have B proteins only on the RBC. So you could be what? Big B, or BO. Okay, you also could be AB blood, and you have A proteins and B proteins on the RBC. So you could only be AB because you have to have the gene for both A and B to make those proteins. And then you could be type O blood. This is no proteins. So the only thing you could be homozygous O. So going back to this, the A allele is completely dominant to the O allele, but codominant to the B allele. And I could say the same thing. The B allele is completely dominant to the O allele, but it's codominant with the B allele. You might be asking yourself, like I did, well, how about the flowers, the red rose and the white rose? Why is that incomplete dominance? Why is, in this case, A and B are not incomplete dominance, but codominance? You get a mixture when you have a heterozygous like A, B. You get a mixture with both the A and B proteins forming. And you get the same thing with a rose. For the incomplete dominance example that I just gave you, if you have both the red and the white allele, you're going to make the red protein and the white protein, and that's going to show up as pink. And when you have blood cells, red blood cells, if you have the A allele and the B allele, you're going to produce the A protein and the B protein, and you're going to have red blood cells with both A and B on it, just like you have flower petals with both white and red pigment. So what's the difference? Well, the difference is this. The A allele and the B allele are codominant with each other because both of them are completely dominant against the O. So that's how picky it gets. So if you have a recessive allele like O, well, we don't draw it like a little o, we just draw a capital O. But if you have a recessive allele and you have two codominant alleles that are completely dominant to that recessive allele, then you have what we call codominance. I don't see the big difference. I think it's just too picky. Okay, so you should be able to do crosses like this. If dad's blood type is AB and mom's type is A, what is the chance that a child would have type A? Well, me saying that mom's blood type is A, it could be, the genotype could be AO or AA. So you're going to have to do two crosses and then multiply the probabilities. So type A would be any kid that's AA or AO. So let's do it. So dad is AB, so I'm going to segregate his genes up here. Mom's type A, so I'm going to do either. So what if she's AA? I'll do that one first. AA, AB, AA, AB. And what if it's like this? AA, AO, AB, BO. Okay, so what's the chance of having big A? What's the chance of getting A something else, but not B? So I should be careful about that because we have codominance. So what's the chance of getting big A, or big A? Oh, so here we got 1, 2. That's it. So 1/2 of it, 50%. What's the chance here? We got of getting AA or AO. Okay, we got one here. We got one here, but I don't count the others because they're not type A. So that'd be 1/2. So I take those two probabilities. Now, because it's an or situation, it's not and. I mean, you're not going to have this cross and that cross happening at the same time. So you say 1/2 or 1/2. So basically 1/2. 50% of the kids are going to be type A blood. Now, you could look at this whole thing and say, okay, I have 4 out of 8. Maybe that's easier. So that'd be 1/2 of all the kids that they're going to have has a chance of having A blood. Now, what if I ask you, what's the chance? Okay, that's part A. What if I ask you part B? What is the chance of them having A B O kid? So you go up here and you see, okay, there's 0% chance there. And then there's 1%, 1 out of 4 here. But since we don't know that the mother is AO, she could be AA, we'd have to look at the whole picture here. So it'd be 1 out of 8 would be my answer. Okay, pleiotropy is a special case where one gene can affect many different phenotypes. So it can affect many other traits, usually in a bad way. So sickle cell anemia, for example, is a pleiotropic gene relationship. That one gene for sickle cell anemia will change a lot of different traits. If you had a sickle cell gene, what happens is the cell becomes crinkled up. It looks like a sickle under low oxygen conditions, like you're exercising really hard. Okay, when these cells sickle, they clump up and they clog tiny blood vessels, causing pain and fever. Could possibly damage nerves, your brain, and damage heart muscle. Also, it can damage your spleen. So all these damaged tissues are different phenotypes from the healthy phenotypes of healthy spleen, healthy heart muscle, healthy nerves, no pain in our joints. But so one gene affects all kinds of phenotypes, such as damaged tissues and organs. Okay, sickle cell anemia. I don't really know what letters to use. I'll use A, little a. So this is the sickle cell anemia allele. Okay, so if you have little a, 2 recessive alleles, you have sickle cell anemia. Under low oxygen conditions, you will suffer with all the traits we just talked about. What will happen is this causes the little a allele causes a change. One little change in the amino acids in the hemoglobin molecule in the red blood cell, causing it to drastically change its shape and to cause the whole red blood cell to crinkle up until you get more oxygen. that causes all the organ and tissue problems that I talked about. If you are heterozygous, big A, little a, you produce both normal cells and a few sickle cells under low oxygen conditions, but you won't feel any effects. So you don't, this would be sickle cell anemia, you don't get sickle cell anemia. And if you're homozygous big A, of course you don't get SCA. So this could be, little a could be considered to be like codominant with big A, but it's not like it's equal. The little a hemoglobin is being made at the same rate that the big A hemoglobin's being formed. You only get a little bit of sickle celling under low conditions, and it's not noticeable to those people that are heterozygous. So, I don't know. This shows variation from what you learned about with complete dominance. Okay, we just talked about pleiotropy or pleiotropic inheritance. Now we're going to talk about polygenic inheritance, which is just the opposite of pleiotropic inheritance. In polygenic inheritance, many genes are involved with a given trait. For example, your height's not determined by one gene, it's determined by many genes. That's what poly means. So, it's an example of polygenic inheritance. Many genes act to produce a phenotype. It's different from pleiotropic inheritance, where one gene can affect many different traits. Another example of polygenic inheritance would be our skin color. There's at least 3 genes that determine the color of skin, and there's probably more than that as well. Okay, the point I want to make is that for polygenic inheritance, you'll get a whole range, like a bell-shaped curve in a big enough population of the heights across the population. So, for example, let's look at this curve. The women. Okay, there's only a few really short women. Then most women are right around 65 inches tall in the population. There's a whole bunch that are like 66 or whatever, 62 inches right here, kind of an outlier. And then the tallest women there's less of them as you go. So it's a bell-shaped curve. Here's the average at the peak. Men, the average is higher than women. And the maximum height of a man, is bigger than in this population than the maximum height of a woman. Okay, but so if we go to the next, this is just showing us that each of these 3 skin color genes has a it's like codominance, but they say incomplete dominance, whatever. You have a light version and a darker version. So big A would be dark. Big A, little a would be medium dark. Little a would be very light. Now, but there's 3 genes like that to do the same thing. So BB, same thing. CC, and there's probably more too. Different kinds of pigments in the skin. So again, this is polygenic, many genes determining the skin color. The relationship between the big letter and the little letter in each of these 3 or more skin color genes is incomplete dominance. There's a blending of the pigment formation of both alleles. Okay, so we're not going to do any crosses with that, but this next picture shows, I don't know if you can zoom in on it, whatever. It is showing the distribution in skin pigmentation or skin color amongst a population of people. And you can see that there's a bell-shaped curve with most people in this population having that color skin. Of course, environmental factors affect your skin color. You know, any exposure to the sun, you'll get darker skin. Unless you're very pale, then you'll get sunburn. Your diet also influences your skin color to a certain extent. So you'd have to have all the people in the population exposed to the same environmental factors to really study that. So many traits are a combination of heredity and environment. What you get from your genes and what you get from your environment. So obviously, if you're malnourished, you're not going to grow to your full height potential. So your genes are not going to help you if you're not given the vitamins and nutrients. Your genes aren't going to help you attain the height that the gene is coding for. Okay, so here's some questions. In snapdragons, heterozygotes have pink flowers. Heterozygotes have pink flowers. Okay, so let's do white and red. Since you have pink, it's going to be what? Incomplete dominance. So R is red. Well, R is red. Little r is white. And homozygous is pink. Again, this is incomplete dominance. You got blending. When plants with red flowers are crossed with plants with white flowers. So here's the red with white flowers. Okay, so let's do a Punnett square. And you can see that it'll be 100% pink. What percentage of offspring will have pink when red is crossed with white? Okay, so 100%. 1, 2, 3, 4 out of 4 are pink. Okay, same thing. When 2 plants with pink flowers are crossed, okay, so we're going to get 1 to 2 to 1 ratio. What percentage also have pink flowers? 50% pink. People who carry a sickle cell allele on 1 chromosome are heterozygous for a sickle cell allele. Well, if they carry it, one chromosome, they have two chromosomes, right? So let's say that this is, I don't know, let's do AA, where this is normal hemoglobin, and little a is sickle cell hemoglobin. So if you have little a here, you could have on a homologous chromosome, you could have big A, and then you'd be okay. So you could be healthy. Okay, so are heterozygous for the sickle cell? Yeah, you could. So I think what they're saying on only one chromosome, we should emphasize that. So that means the other one would have to be big A, because it said only on one chromosome. So they're heterozygous, big A, little a. They're healthy. They won't feel the effects of sickle cell anemia. They produce both normal and a little bit of abnormal hemoglobin. So really it's all of the above. Which the following is a direct effect on sickle cell allele. It's a crinkling of the hemoglobin molecule under low oxygen conditions. So really it's because hemoglobin, the crinkling is not the direct effect. The direct effect that causes all the other problems is that you made some hemoglobin, well, you made all your hemoglobin into a different shape, abnormal, so that when your oxygen conditions are low, the hemoglobin crinkles up. Okay, you got the same genotype of these two flowers. Some plants produce blue flowers and others pink flowers. What? Okay, so when they're crossed, they have the same genotype. Okay, if they have the same genotype, it's not the genes that are causing the color differences. It's environmental factors. It could be something in the soil. Actually, it is, if you know pH paper, litmus paper, maybe use that in grade school. It turns blue in bases, it turns red in acids. Well, that's the pigment in the petals of hydrangea plants, flowers. So if you're in an acidic soil, you'll turn pink. You'll produce pink flowers. And if you're in a basic alkaline soil, you'll turn blue flowers. So environmental factors such as soil. Okay, polygenic means many genes produce the trait. So they're the bell-shaped curve distribution of different traits from 2, 3, 4 or more genes, polygenes. Okay, so not this, not that. That's incomplete dominance. This is codominance. I don't even know what Huntington's disease is, but the skin color, I do know Huntington's, but that wasn't even discussed. Skin pigmentation in humans. Skin color, so that's it. So it would be height. Okay, errors in replication, DNA replication. Can lead to mutations if they're not repaired in the DNA. If the error is to the DNA in the germ cells that makes the germ cells that eventually do meiosis to make the gametes, only those kind of mutations can be passed on or inherited by the children. So we talk about mutations. Mutations can happen to our body cells and to our sex cells. Okay, but unless they act on our sex cells, you're not going to pass on the changes, the mutations to your offspring. There's all kinds of genetic disorders. We talked about a few of them for when the chromosomes mess up, do something different in meiosis than they're supposed to do. But that's only with the chromosomes in our germ cells, because those can be passed on to the kids. So a lot of the disorders, the genetic disorders or genetic diseases, are due to mutations that cause a trait that's harmful. But it's a basic complete dominance kind of thing for most of these disorders. Many of these genetic disorders are due to recessive genes, a completely dominant recessive situation. For example, if you have big A, you're healthy, you're not sick. If you have little a, you're sick. And if you have big A, little a in your diploid cells, I'm talking about, you're healthy. But you carry the recessive gene. But if it's complete dominance, you miss being stricken with the disorder, because the big A makes up what the little a's not making. Okay, we call the heterozygous individual, we call that person a carrier, because that person can carry in a hidden fashion the problem recessive allele and carry it on to her kids, his or her kids. Okay, an example of a recessive disorder. When I say recessive, I mean complete dominance. But it's a recessive, the lowercase allele, that causes the problem. So, albinism, where you don't produce any pigments. Your skin, your hair is totally white. Your irises in your eyes are red because there's no pigment. That's the blood vessel showing through. Another example is cystic fibrosis. CF is a horrible condition that it causes you to make too thick a mucus in your lungs. And what happens is your lungs can't expand because of the sticky mucus. And the air sacs in your lung, the microscopic air sacs in your lungs, eventually can't become unstuck and fill up with air. Okay, again, that's due to a recessive allele. If you have, let's say you have this, you're carrying it. You're healthy. You're totally healthy because the dominant makes up for the recessive. It's completely dominant. And even though you being a carrier are healthy, you could pass that recessive allele to your children through your gamete. So this is representing cystic fibrosis gene, the little a. If you have two heterozygous normal people, they could get a child with cystic fibrosis. Okay, so there's a 25% chance if two carriers mate, two normal non-cystic fibrosis people, but two that are carrying the gene, the allele for cystic fibrosis. If they mate, there's a 1 in 4 chance, 25% chance of getting your child, your newborn child, having cystic fibrosis. And there's a 50% of producing a new carrier, so it keeps the little a allele, the harmful allele, in the population. So if I were to ask you, two carriers mate, what's the chance of their child being healthy? So each time they have a child, there is how much of a chance that they're healthy? Well, 75%, right? 3 out of 4. But there is a quarter chance. There's a 25% chance that any kid that's produced by two carriers would have cystic fibrosis. Inbreeding can cause rare recessive alleles to build up in that family. So much more probable that recessive allele will build up and remain in a family line when there's inbreeding. That's like when your brother and sister they mate, or a father and his daughter. Sounds pretty sick, but it does happen with people, or a son and the mother. And this is why, like, purebred dogs and animals sometimes have rare diseases that make them very unhealthy in several ways. The mutts seem to be the most healthy. So in Kentucky, in the mountains of Kentucky, I guess, you got little tiny communities, and there was inbreeding in the community, and there's this very rare gene that causes your tissue to turn blue. This lady's face is bluish. And that's because the red blood cells are having a hard time of carrying oxygen. And so, not because they're sickle, just because something in its shape is not allowing it to effectively absorb as much oxygen as it should. So the blood is like vein blood, even in the arteries. Okay, so that trait remains in that population of people because of inbreeding, whereas it's very rare in the larger population. Okay, here's other examples. Even sickle cell anemia is a recessive disorder. I kind of think I kind of think of it though as a, like I said before, as an incomplete dominance issue, but really that's kind of iffy because you don't form equal amounts of hemoglobin, normal and abnormal hemoglobin. If you're AA, you produce much more normal, so you don't even feel like you have the disorder. Tay-Sachs disease, another horrible disease where fat globules grow, build up in your brain, and the baby in early childhood loses his intellectual, his brain activities, and it's horrible to see your child, of course, go slowly brain dead. Now, there's some, don't think that all genetic disorders are recessive. A lot of them are. But some are due to dominant alleles. So one form of dwarfism is due to a dominant allele. Now, if you have both dominant alleles, that will kill you. You won't develop with this type of disorder. But if you are heterozygous, you'll be a short little guy like this dude right here. But it won't kill you. And if you're little a, your normal height. Okay, if you're heterozygous, you're a dwarf. And if you're homozygous dominant, you're dead. It's a lethal situation. Okay, Huntington's disease is another dominant genetic disease due to a dominant allele. You can't tell you have it phenotypically until you get to middle age, and then your nerve function just starts degenerating. The sad thing about that is you can have kids and pass it on to your kids without knowing that, you're being a parent, that you did that until you start getting the symptoms in mid-age. Okay, so Huntington's disease is caused by a completely dominant allele. So you could have Huntington's disease if you were like that or like this. But, so if one of your parents has a disease, so it could be big A, or big A, little a. And the other parent would have to be homozygous recessive. So what's the chance of you getting it? Well, if your parents big A, chance of you getting it is 100%. But if he's heterozygous or she's heterozygous, it'd be it'd be 50% there. So, because this is an or situation, we don't really know which way the sick parent is. What we do is count 4, and then 2, so it'd be 6 out of 8, or 3/4. 1, 2, 3, 4, 5, 6, 7, 8. So 75% chance that you'll get it too, which is pretty high. No, it would be, it would be only 50% if the affected parent was big A, little a. So either 100% or 50%, or take the average of those, 75%.